This video will introduce several applications of nuclear magnetic resonance spectrometry, or so-called NMR. It will introduce fields that widely use NMR, like identifying compound structures, analyze complex components, and medical imaging. We'll talk in detail about applying NMR into understand ligand binding, which is applied to drug discovery and its effectiveness testing. NMR is a good way to identify organic compound structure. Organic compounds' molecular formula can be identified using other methods. However, several structures may all have the same formula. For example, for the compound that has 9 carbons, 11 hydrogens, 1 nitrogen, and 2 oxygens, these are several possible structures. To determine the structure that corresponds to a compound, we can use NMR to identify different hydrocarbon groups. And in this case, given the NMR, we can conclude that the compound is benzocaine. NMR has also been used to fields that need to analyze complex components. For example, it has been used to understand the brewing process of beer. A group had used NMR to understand the components of beer during the whole brewing process, and they are hoping to use the result to further improve the quality and taste of beer. An important aspect that applies the principles of NMR is magnetic resonance imaging, or so-called MRI. It makes it possible to separate tissues with different properties, and the imaging result can help doctors in the diagnostic process, for example, to identify possible tumors. NMR had also been applied to understand ligand binding. To better illustrate this application, we'll talk about analyze the effectiveness of different drugs that attacks human immunodeficiency virus, or HIV. HIV is a representative RNA virus. Its genetic information is encoded in its RNA, and to reproduce itself, it needs help from host cell to replicate its RNA. However, most eukaryotes cannot replicate it. HIV virus uses reverse transcriptase to synthesize DNA, for which will be able to infect the host. To battle against HIV, many drugs have focused on blocking the process of reverse transcription. This image shows the structure of a HIV reverse transcriptase, and it is one of the most important enzymes that is needed in the reverse transcription process. There are 2 sites that are the most important for its functionality. One is the polymerase, and the other is the nuclease. Single-strand viral RNA is synthesized into a RNA-DNA hybrid by the polymerase, and then the nuclease broke the hybrid into a single-strand RNA and a single-strand DNA, while the single-strand DNA will further synthesize into a double-strand DNA and be combined into the host cell's genome. To block the reverse transcription process, 2 kinds of drugs had been developed. The first type is called nucleoside reverse transcriptase inhibitor. It will be attached to the RNA, which will block the reverse transcription process. The other type is called non-nucleoside reverse transcriptase inhibitor. It will bind with the reverse transcriptase, which will be able to block the reverse transcription process. When the DNA-RNA hybrid cannot be synthesized, we have a way to block the replication of HIV virus. However, different non-nucleoside reverse transcriptase have different shapes. For one specific drug with specific conformation, it may bind tightly to the reverse transcriptase, which is shown in the simple demonstration. But for a drug with a different conformation, the binding may be a lot weaker. In this case, a higher concentration is needed to achieve the same effectiveness. Normally, the 50% effective concentration is used as a metric to measure how effective is a drug. Drug binding is a process where chemical bonds are formed and will cause chemical shift, which can be well measured using NMR. A group of researchers from Thailand and Japan tried to use NMR to find a metric that measures HIV drugs' effectiveness. They labeled important amino acids on the reverse transcriptase with carbon 13, which makes those amino acids identifiable on the NMR spectrum. They first measured the original reverse transcriptase two-dimensional NMR spectrum, which is shown on the left. Then they added nevirapine and measured the spectrum again, and some of the amino acids had shifted on the spectrum, which may likely be due to binding. Also, they found that some amino acids, like M230 in this case, are made soluble after nevirapine is applied. Another thing that they found is that different drugs would cause different amount of chemical shift. In this case, different amino acids shift different amounts when the applied medicine is either nevirapine or ripivirine. Based on their experiment result, they found a strong correlation between the chemical shift of a specific amino acid, which is M230, and the effectiveness of different drugs, which is quantified by the 50% effective concentration. They propose that future HIV non-nucleoside reverse transcriptase inhibitor drug screening can be done by using NMR and measuring the chemical shift between different medicines. In this video, we have described several applications of NMR, which include identifying compound structure, analysis of complex components, medical imaging, and ligand binding analysis.